Hello, my name's Alen Faze, and today I'm going to be doing a single cell sequencing workshop focusing on cellular deconvolution. So this all stems from the recent publishing of the Human Lung Cell Atlas, or HLCA. This atlas involved taking 14 data sets of single cell data from around the world and making a core atlas, basically a resource where single cell sequencing data has been integrated to allow for the identification of potentially new rarer cell types and where particular genes are expressed. Now, the core atlas is substantially smaller to the extended atlas, which involved 49 data sets. This involved a lot more diseased samples and about 2.4 million cells. What's important to realize is this extended atlas is so big that many people are unable to actually load this on their computers. So a lot of people actually focus on using the core atlas for most research purposes. However, you can use a publicly available web server that allows you to integrate, to investigate these data sets quite easily. And let's just quickly show that now. Here you can see the lung atlas, you can see the full data set and also the core data set. Here we allow you to download these data sets, but again, I warn you that the full data set is extremely large and not many computers can actually run it, or servers can run it, while the core data set is still large, but you should be able to run it on a server at your university. Now, what you can do is you can press Explore. This will open a new tab and basically allows you to investigate the data set on a web server. It does take a little while to load, as you can see here, but what you can see is a UMAP and a number of different variables that allow you to do different comparisons within these data sets. What you can do very quickly is that you can look at the different types of cells that have contributed here. You can label them here. You can see the labels here if you click. And here what you can see is all the different cell types that have been identified in the atlas from all the different cohorts. You can then type in particular genes, for example, IL-33, and then look at their expression profiles by pressing this color tab here. And what you can see is that the IL-33 is mainly in the resting basal cells and also in the endothelial cells that you can see here. Now, what's important to remember is that these images are of sufficient quality to be published. It does so making that you don't need coding experience to use single cell data in your current publication. The other thing it allows you to do is do differential expression between samples. For example, what you can do is you can go down to smoking status, and then you can compare active versus former smokers. So for example, you hit active. Now there is a limit on the number of cells that you can compare against each other. We might have hit this here, but basically here we can compare active and former smokers to see which genes are differentially expressed between them. This also allows you to do it within cell types as well, so you could just focus this on mast cells. However, at the moment, we're doing this across all cell types in former and active smokers. So we'll give this a few moments to complete. While that is running, what you can also do is the lung atlas is contributed to both from nasal, bronchial, and also parenchyma. So what you can do is check the tissue levels, and you can select only the tissues that you might be interested in. So you've got parenchyma, only trachea. So that way you can only look at particular cell types that you may be interested in. So our differential expression analysis is finished. And now what you can see, here are the genes that are down or higher in smokers, while genes that are higher in population 2, former smokers, can be seen here. Okay. Well, that's all I wanted to show you on the lung atlas. Let's go back to today's task, and what we really wanted to do is focus on the cellular deconvolution. Okay, so, but first we need to know a little bit more about basic RNA sequencing. So it's important to remember when we normally do sequencing, we use this on bulk tissue. So this involves taking a tissue sample and then doing bulk sequencing on that, and basically involves taking a tissue sample and grinding it down to the point that it is all mixed together. And then you basically get a single signature or single value for a gene across all those different cell types that were in that particular sample. It can be thought of as a fruit smoothie, where you can't taste each individual fruit, but you know sort of that each fruit is in there, but you don't know the contributions or how much each fruit is contributing to the overall flavor. Same with gene expression. Although a gene might be differentially expressed, you don't know if it's coming from the basal cells or mast cells. Well, with single cell sequencing data, each individual cell is sequenced, and this can be thought to be a fruit platter where you actually get the expression from each individual cell, so you can identify which cells are actually expressing a particular gene. So how does cellular deconvolution come into this? Well, cellular deconvolution is important because we know that when we collect a sample from a patient, such as a bronchial biopsy, they're not all the same composition. Many will have more basal cells, more ciliated cells, or more airway smooth muscle. So what we can use is a single cell reference. Here, we're going to use a single cell lung atlas and basically deconvolute or re-identify these cells or the contribution of these cell types from the original sample. For example, we can see here we've got much more of this red cell compared to the other samples. So we can do this for the lung. This has been previously done, focusing on the lung tissue atlas. Basically involves looking at 1,000 individuals using microarray analysis. This was then deconvoluted using the single cell lung atlas matrices that we'll be talking about in a moment. And this allows you to determine how much of each cell is there, but more of proportion, and allows you to compare between diseased and non-diseased. Here, they didn't actually find anything to be statistically significant. However, they did run into some problems. And what is it you do need to be careful of when you're doing deconvolution? You can do it on microarray data, but if you use some really old microarrays, some of the old AFI microarrays or Illumina microarrays, where they actually measure a gene multiple times, you do need to pick the best probe set for each gene. Otherwise, you may get quite strange results. But not all cells are deconvoluted equally. What's important to realize that both capillary endothelial cells and interstitial macrophages that from the airway, although they were included in the deconvolution matrix, they have very poor performance during benchmarking. However, their addition to the deconvolution actually greatly improves the accuracy of all the other cell types. So this is important to realize, that they're in the matrix to improve the other cell types, but they themselves should not be looked at in the airway when you do deconvolution of airway samples. It's also important to remember the platforms. So for the nasal and airway matrices for the deconvolution, they are using ensemble IDs, while the lung tissue matrix is using gene symbols. Cell types that cannot be detected in large number of samples should most likely be excluded from the analysis. For example, if you're looking at 100 patients and you find you've got 95 of them have 0 of a particular cell type, only 5 have that cell type, it's best not to do any statistics because you will get inflated p-values. So please make sure that the cell types that you're looking at don't have too many zeros in them. So the workshop today can be accessed very easily at the following link, the RBMB-lab.com. So let's go there now. So you'll come to a page such as this. All you need to do is go to workshops and then go to the cellular deconvolution workshop. And it will take you through step by step the processes of what we will be doing today. Now, it does have a bit of information about the lung cell atlas or single cell lung cell atlas, and then how to proceed and actually do the deconvolution. So the deconvolution matrices from the lung cell atlas paper are available in supplementary table 10. However, we've also made it available. If you just click this link here, it'll allow you to download them directly. Now, these are zipped, so you will need to unzip these and put them on somewhere onto your desktop. Now, for today, for the example, we will be taking a data set that was previously published under the GSE number of 178807 and can be accessed here. So you can download this very easily. We've made a subset of this so that the deconvolution will run quite quickly, allowing you to do some quick tests to get the deconvolution working. Once you've downloaded both these groups of files, then you just need to go to CIBERSORTx, click this link. So with CIBERSORTx, you will need to log in, make a login, but once you've logged in and come to the main page, you should see this Run CIBERSORTx come up. And then what you will need to do is go to upload files. So there's 2 different types of files that we'll be uploading today. These will be the files that you will be using for today's analysis. So the ones you've just downloaded. So what you'll need to do, I've already got my files ready. So we're going to be using the airway signature, type signature file. This has been uploaded here. We can just call this CIBERSORT airway. And then we will call this a signature matrix. And then you can upload the data. And then what we'll do is we'll add the mixture file that we've created. So this is your RNA-seq file. We call this RNA-seq. And this will be our mixture file, and we can upload it there. Now, we'll go through a little bit some of the aspects of this file after, but first of all, let's get this up and running. So then all we have to do is go to Run CIBERSORT, go to option 2, Impute Cellular Factions, go to Custom. Here, for our signature matrix, we're going to go to our CIBERSORT airway. And for the mixture file, we'll go to our RNA-seq file as well. Leave everything as standard unless you're using microarray data. We don't have to do any batch corrections here because these were all sequenced at the same time. We don't need to we want to disable the quartile normalization because this is RSX data, and we don't want to run in absolute mode at this point in time. Now, usually what you would do at least 100 permutations to run your permutation analysis. But for the sake of time, we're just going to run it with none. But make sure when you do it for yourself, you set it 100. It may take about 20 to 30 minutes to run. If you set it at none, it will only take about a minute to run. So while we're waiting for this to run, what we'll do is we'll just have a look at the file, the matrix file that we were looking at. Just give me a moment while I get that up and running. This was a mixture file that we just did. We've got 5 healthy and 5 COPD. We can see that this is a tab delimited file, so these are separated by tabs. There are 2 things that a lot of people forget when they're making their matrix files and the main errors. You must give the ensemble ID column or the gene ID column, depending on which matrix you're looking at, a name. Doesn't matter what it is, but it needs a name. Otherwise, you will get an error. In addition, you need to know which matrix you're using. If you're using the nose and airway, you need ensemble IDs. If you're using the lungs, you must use gene symbols. Okay, let's go back to our analysis and see how we're going. So if we go back to our analysis, we can see it's running quite well. We're about 50% of our samples deconvoluted. And basically, this allows, you can see this in real time. You do not need to keep your web browser open. You can always check this later. But if you're just running that sample for this test, it may as well just have it running in the background. So once this is completed, you will have a nice file that you can download, and then you can do associated statistics with this. You can basically do t-test or Mann-Whitney U or Wilcoxon, depending if you want non-paired or paired analyses. Just be careful with zeros. If you do have a substantial amount of zeros, again, you probably want to do a non-parametric test. Okay, here we can see this is now completed. We've got our healthy individuals, we've got our COPD individuals, and then we've got the cell types. Remember, you should not disregard any analysis that comes out of the EC capillary and also interstitial macrophages, as these data is not accurate. Now, this was brushing data, so we're expecting mainly multiciliated and basal secretory, and that's pretty much what we see. This is quite nice. Now, we could compare healthy and COPD here. Now, what we are mainly seeing is that although it's not significant, we do see sometimes a higher proportion of basal and secretory cells in COPD patients than we do see in healthy, but this is not always the case. You probably need a greater N if you want to see statistical significance. So that was it for today's workshop. It was really there just to show you how to run the analysis. That table you can just download as a CSV file and run it in any statistical program that you would like. Now, it's important to remember this resource will remain on the RBMB-lab website or GitHub for the foreseeable future, along with a link to this workshop as well. I'd like to thank you, everyone, for joining. We will be doing other workshops in the future through Symbol at TZANS. So keep an ear out, and we will advertise through TZANS if you are interested. If you are interested in joining us, just shoot me an email, or just get your colleagues to send you the links. Thank you again for joining us. I'll catch you later. Bye.